Parts of the microscope includes four systems: supporting system, illumination system, magnification system, and adjustment system. So this is
So,
monocular.
which are the four systems that you need to know about the microscope? Important four systems. That is the first one. Yes, supporting is the first one. Which are the other three? Which are the other three system of the microscope? Very good, very good. Okay, we proceed.
Compound light microscope. Supporting system. In the supporting system, we include base. This is the pillar, handle of the microscope. This is the body tube. This is the revolving nose piece. The structure which is rotating, it is known as revolving nose piece, and here it is the mechanical stage of the microscope. Mechanical stage is the stage on which we have to put the slide or the Newbar's chamber, and we can move this mechanical stage either forward or backward and from right to left side. Illumination system. In the illumination system, here it is artificial yellow light bulb is present in this black color structure. This is the screw. Whenever we rotate this screw, whatever the structure is moving up and down, it is known as condenser. Now you are clearly seeing this. This is the structure which is moving up and down. It is known as condenser, and there is a one pin which is present and through which we are going to regulate the iris diaphragm or the openings of the iris diaphragm. So this pin, whenever we put it on one side, the iris diaphragm is completely closed, and whenever we push this pin on the opposite side, the iris diaphragm will be completely open. So this is the pin to regulate the amount of the light rays which enter into the object, and this part it is known as condenser. Alright. What is the use of condenser? It condenses rays of light and focus it to the object. And what is the use of iris diaphragm? It regulates amount of light entering the object. It is the light microscope. So here you have to insert 3 pin inside the plug which is present over the switchboard, and you have to on the switch. Once you on the switch, there are also the manual setting are present on the bottom part of the microscope. Here on off switch, and here low and high illumination, or the whatever the whenever we put this on the low, there will be the darkness of the vision, and whenever we put this high, there will be the brightness of the vision. So here we can manage on off as well as low and high vision or the brightness of the vision. Magnification system. Inside the magnification system, there are two convex lenses are present. One, here it is the eyepiece lens, and here it is the objective lens. Here we are using eyepiece lens of the 10x power, and in the objective lens there are three types of objective lens we are using: 10x, 40 or 45x, and 100x. 10x lens is also known as low power, which are using for the
All of you should remember, when you come to the practical class, pathology practical class, you will be using microscopes. So the microscopes, some of them have 4 objectives, and the first one is 4x, 4x, and that is known as scanner. Then we come to 10x, and further 40x and oil immersion. So 4x is, remember, scanner, which is placed along with the 3 objective lenses.
WBC count practical. 40 or 45x lens, which is also known as high power, and we are using this lens for the RBC count practical, and 100x lens, which is known as oil immersion lens, and this lens we are using for the blood smear examinations. Whenever we are putting objective lens as a 10x, total magnification will be 100 times. Whenever we are putting objective lens as a 45x lens, total magnification will be 450 times. And whenever we are using 100x as a objective lens, total magnification will be 1000 times.
So how you
Times.
come to the calculation of total magnification? That is just the magnification of eyepiece multiply by magnification of objective lens. So this is again very important. Total magnification is just multiplication of magnification of eyepiece lens and objective lens.
And whenever we are using 100x as an objective lens, total magnification will be 1000 times. Now the adjustment system. Inside the adjustment system, there are two adjustment system are present, coarse adjustment and fine adjustment. The bigger one screw, it is known as coarse adjustment screw, and this smaller screw, it is known as fine adjustment screw. When we move this big screw or the coarse adjustment screw, you are able to see the movement of the mechanical stage, which is perceived by the naked eye. So the movement is occurring in the centimeters when we rotate this coarse adjustment screw. When we rotate this fine adjustment screw, the movement occurring in millimeters. So here we are not able to perceive the movement of this mechanical stage via the naked eyes, but we can observe or we can perceive the movement of the mechanical stage from this eyepiece lens. How you are going to identify the different types of objective lenses. On the objective lens there is a written 10x, 45x or 100x, or simply they will colored. Yellow color is for the 10x, blue color is for the 45x, and white color is for the 100x. Now this is the table in which I am going to explain the position of condenser and iris diaphragm in the different type of objective lenses. 10x lens, which is also known as low power, it is used for the WBC count, and the position of condenser, it is the lowest, and iris diaphragm partly open. 45x, which is also known as high power, and it is used for RBC count, and the position of condenser midway between the lowest and highest, and iris diaphragm is half open. 100x, or it is also known as oil immersion lens, which is used for blood smear examinations, and position of condenser highest and iris diaphragm fully open. So these are the settings whenever we are using 10x lens, 45x or a 100x lens as a objective lenses. We have to change the position of condenser and iris diaphragm according to their power.
So here you have come to know about the use of the objective lenses, that is only for hematology. But in our further practical class, that is now we are discussing today, you will also come to know where these objective lenses are used apart from hematology practical class, that we will discuss further in the next few slides.
As objective lenses, we have to change the position of condenser and iris diaphragm according to their power. This is the objective lens, and here on the mechanical stage we are putting some of the object over the slide. Here object means either the cell or different types of structure. And whatever the distance between this objective lens and our object, it is known as working distance. The working distance is highest in the 10x lens. The working
Here
distance is midway.
the working distance is shown as red colored arrow. Okay, very simple to understand. Here red color arrow is there, which you understand as working distance.
Way between the 45x lens, and there is no any type of working distance is present because 100x lens it is known as oil immersion lens. While we are using 100x lens, we have to use the cedar wood oil and immerse the 100x lens inside the oil so that we will get the highest level of resolution. Why we are using cedar wood oil, but here we are not using any other type of oil, because the refractive index of the cedar wood oil is same to that of the glass light, so it prevents the refraction of light rays, and the whatever the image we are seeing from the eyepiece lens having the highest level of the resolution. And I explained about the light microscope. In the simple microscope, there is not any presence of artificial light. There is a simple mirror is there. There are two sides of the mirror. One side it is the plane mirror, and on the opposite side it is the concave mirror. Plane mirror we have to use whenever there is a diffuse source of light or the light is natural, for example natural sunlight. And concave lens we are going to use whenever there is a source of light, it is the artificial tube light. And so this is all about the monocular compound light microscope. I hope everyone understood about the this monocular compound light microscope in detail. And if you like this video, please try to share it with your groups, friends, colleagues and batch. Hello everyone. Today we begins with the topic of microscope, and I am going to explain the different parts of the microscope in detail. Illumination system, magnification system and adjustment system. So this is monocular.
Minimize कर minimum minimize हाँ अधिक minimize अब आम बंद करते बस बस बंद करते हैं ऑफ कर हाँ हाँ Okay, so we have showed you the video on microscope, and that is more than sufficient that you should be learning as a second MBBS student in pathology. So we have shown you the video on microscope, and I think you should have understood all the details of it. Now, my next question is, what is the use of microscope in medical field? Because you are a medical student, so you must know its use in your field, use of microscope in your field. So what is the answer? You have already seen the use of it in one of the section of pathology. So what are the other uses? Can anybody put the answer in the chat box. What are the other uses of microscope? Where do you use it? Okay. Very good. So somebody says it is for the organ, tissue analysis, correct? Organisms, very correct. So you know it where we can use it. So before we proceed to the use of it, because we will be discussing under light microscope. So the types of the microscope, you should just have the idea what are the types of the microscope. You are not going to study the details of it, but only remember you will be studying in very brief. Only some more details that you need to know is for light microscope. So in 1665, the scientist was Robert Hooke, who developed the first laboratory compound microscope. So this one is the diagrammatic view of the microscope, and the parts have been shown in detail in the video. Again, to revise the function of the parts, we will just go through it. The first was the eyepiece, which was of 10x, and you below this eyepiece you have a tube-like structure. So this is the part of the microscope you look through. Then comes the body tube, which connects the eyepiece to the arm of the microscope. The arm itself, it connects to the body tube to the stage that is lower down the arm, and that is the part of the microscope that you hold wherever you are carrying it, or at the place where you are studying something under the microscope, you just hold on it. The stage is where you put your slide. Then comes the objective lenses, that is known as the revolving nose piece, where they are attached. And all three, as I told you, the lowermost is 4x, that is known as the scanner view. Then comes 10x, then 40x, and the fourth one is oil immersion lens. Then comes the diaphragm, that is you find the hole in the stage that allows light to pass through. The base of the microscope, that is the lowermost part on which the whole of the equipment holds, and that is the second part you hold whenever you are shifting the microscope from one table to the another. And later are the fine adjustment and the coarse adjustment screws. So now the next is, you see here the light or the compound microscope, the uses we have already seen in the video. The first is the use in hematology lab, where you study the cells. Here you are studying the WBC cells. You are performing the total WBC count, and this one is under the low power lens, and where you are adjusting the chamber, Newbus chamber, and then under high power, you are observing the cells under the Newbus chamber, and on the rightmost, you find under oil immersion lens, you are studying the peripheral blood smear. So that is in hematology. Next is, you are studying some of the fluids, and the area where you are studying is clinical pathology. So in clinical pathology, when you are study the fluids under microscopy, the most common would be the urine sample. So here we have shown you A, B, C are the urine sediment which are examined under light compound microscope. Next is, you even study another sample in clinical pathology, and that is the stool sample. So stool microscopy also can be done under the same type of microscope. Or even here we have shown you the urine sample. Any of the fluids of the body can be studied microscopically. Second was the stool sample, and the last in pathology is the use in histopathology, where you study the disease tissues. Okay, you study the disease tissues. So these are the uses of light microscopy in pathology. Next, somebody said you can observe even bacteria or microorganisms. Yes, very correct. So in microbiological examination, you can study the bacteria. It may be pus or any fluid which contains bacteria. So the use is very, important when you can observe the organisms under the microscope. Now we will see very brief details of the rest of the microscope. Okay, very brief details of the rest of the microscope. Let me check box. ये लोगों ने चलो
So now
you see about the other type of microscopes, just the principle and one or two uses of that microscope.
So now we will discuss in brief about the phase contrast microscope. What is the principle? A ray of light consists of waves traveling in a straight line. If two such waves travel in the same line, they are said to be in phase and help to produce a brighter image. This phase contrast microscopy uses this ability of waves to help and hinder the light. It is obtained by placing in the condenser an annulus, which allows only a ring of light to pass through, and by placing a phase plate in objective, which has a circle that matches the ring of annulus. So basically, the thing which is apart from the light microscopy is first, as we have seen here, that one annulus is placed near the condenser and another annulus which is placed in the objective. So they are creating a phase contrast by which we can see the small organisms and the cells even without staining them. So this is the working principle of contrast microscope. Here we can see that there are four types of light scattering which is used here: illuminating light, background light, specimen scattered light, and foreground, that is the combination of the background and the scattered light. Now here this is explained that how the vector length and direction respectively corresponds to the light intensity and phase difference, and thereby creating the image. The same cell image is here we can see that with the traditional bright field microscopy on the left side and with the phase contrast microscopy on the right side. In bright microscopy, there is no phase contrast, so we cannot clearly see the detail of the cell. But in phase contrast, because of the this contrast phase differentiation, we can see the detail of the cell. Now, where this is used in bacteria like Vibrio cholerae, Leishmania in blood, Trichomonas vaginalis, amoeba in feces, and urine sediments, where we don't have to stain the specimen and we can see them as it is. Another is the dark field microscopy. Principle is a special dark field condenser is placed, which has a central blacked out area. Uses of the dark field microscopy are in the microbiology laboratory, especially for the following purposes: for the visualization of spirochetes such as Treponema pallidum, which are causative for the syphilis, Borrelia burgdorferi, for which are causative for the Lyme borreliosis, and Leptospira interrogans, which causes leptospirosis in clinical samples. Observation of microbial motility, like tuft of bacterial flagella that can often be seen in unstained cells by dark field or phase contrast microscopy. The differentiating feature between the dark field and the bright field microscopy here is shown that in bright field microscopy, there is condenser lens, specimen, and objective lens, and there is light source. So the full light which is imparted on the condenser lens is passing through the specimen, and we can see the field. Whereas in dark field microscopy, the light is filtered, not exactly filtered, but is obstructed, and it will pass through a small annular like ring, and that will be imparted on the specimen, so that there will be scattering, and against the dark field the object will get illuminated, and we can see the detail of the object. Now, what are the uses of the dark field microscopy? They are used to see the structure and the motility of the organisms like Treponema pallidum, Leptospira, Microfilaria, and Vibrio. One more thing I want to tell you, that the difference between the phase contrast microscope and the dark field microscope is that in phase contrast, there was two type of the annular are used in phase contrast. One is the condenser annulus, which is obstructing light and allowing it to pass through a small annulus, and another gray ring which is used, where we can see that this gray dark ring is just not allowing the light to pass, but other area is allowing to pass the light, so that the phase contrast will be created. Whereas in case of dark field microscopy, only one annular ring is present. So that is the mechanical basic difference between the dark field and the phase contrast microscope. Now come to the fluorescence microscopy. The principle is white light is composed of 7 different colors in the visible spectrum of wavelengths. Other wavelengths which are not visible to our eye are ultraviolet light with very short wavelength. Now, practicals or microorganisms are previously stained with fluorescing dyes, and which transform the visible ultraviolet light into the visible light by increasing their wavelength. This enables the particles or the microorganisms to be seen glowing, that is fluorescing, against the dark microscope. So here we are using the stain, fluorescent stain, and the microscope is enhancing the stain by increasing their wavelength, so that they will be seen by naked eye. Now, where this is used, it is used for the acid-phi bacilli in sputum and CSF, that is cerebrospinal fluid, gonococci and meningococci, Trichomonas vaginalis, and immunodiagnosis. Immunodiagnosis, what is that? Immunodiagnosis means when we want to uh, see some cells which are having a specific antigens, then we can stain them by the fluorescein dye, uh, and we will do this by the antibodies against that antigen. How to see that? For that, we have to stain this antibody specific for the antigen, and that antibodies stained with fluorescent dye will get attached to the antigen, and when we see it under the microscope, we can come to know that whatever we are seeing is the specific antigen against that antibody, so that by this fluorescent dye, the immunologically we can find a cell. Yes, and specific in case of patient's serum. So the antibody antigen reaction will help us to eliminate the cell. Now, next is the electron microscope. Electron microscope uses a beam of electrons rather than the visible light. Electron microscope are used to investigate the ultrastructure of a wide range of biological and inorganic specimens, including microorganisms, cells, large molecules, biopsy samples, metals, and crystals. Here also in biopsy, three types of biopsy where mainly it is used are the renal biopsy, lung biopsy, and skin biopsies. In renal biopsy, electron microscopy, we can see there is extensive fusion of the podocytes and food processes, podocyte food processes. Okay, so now we will go for the questions. First question, a light microscope is also referred to as
Now we will proceed with some of the questions. Back. Now please you listen. If you are going to put any other comment in the chat box, then we will stop our discussion on this practical demonstration of microscope. So please don't put any other comment in the chat box. Now we proceed with the questions. So if at all you know the answer, you please put them in the chat box only. Don't unmute your mic even. So we will proceed. We have here about 20 questions that are related to the use of the microscope only. So we will proceed with that.
okay. Question number 1. A light microscope is also referred to as a someone has that it is known as the compound microscope. Yes. So the compound light microscope is a tool containing two lenses which magnify and a variety of knobs used to move and focus the specimen. Since it uses more than one lens, it is sometimes called the compound microscope, in addition to being referred to as being a light microscope. And in your practical classes, you will be using this light microscope. The second question, the two knobs used for focusing the image include fine adjustment knob and okay, who will answer? Okay, good. The coarse adjustment knob. Here we can see there is ocular lens, which is ocular lens, which is also called as eyepiece. There is a body tube. Then there is a nose piece, which is attached with the three objective lens. Then arm, there is a stage, which is having the stage clips to fix the slide. Then there is a coarse adjustment, fine adjustment, the diaphragm, and the light source. Now the third question is, on the microscope stage, what is used to hold the glass slide in place and prevent it from moving? What is the answer? Yes, the stage clips, which will hold the slide or the specimen in place on the stage. The next question, total magnification is the product of two values, that is power of eyepiece and power of what is the, yes, the objective lens. As the ma'am have told us that magnification of a compound microscope, here we multiply the eyepiece and objective lens power to determine the total magnification. As we have seen earlier that there is a scanning lens of objective lens of 4x, and we have the eyepiece of 10x. So there will be magnification total of 40x. According to that, in low power, when the 10x objective lens is used, the magnification will be of 100x, and for the high power, the objective lens is of 40x. So total magnification will be of 400x. Means we can see the specimen 400 times larger than its original size, and we can see it by naked eye. The fifth question is, which part of the microscope should you hold when lifting or moving the microscope? Yes, which part will you hold to move the microscope? Yes, arm and base. Sixth question is, transmitted light microscope has a light source below the stage, while a reflected microscope has a light source that is located. So reflected microscope has a light source in the eyepiece. In reflected light microscope, vertical illuminator in the light source is positioned so that the tungsten halogen lamp filament is located near the principal focal point of the collector lens. So this is the reflected light microscope. Seventh question is, which of the following is not a type of light microscope? Phase contrast microscope, bright field microscope, electron microscope, or stereo microscope? Yes, answer is the electron microscope. As we have seen that there is electron beam used rather than the light. Eighth question is, the working distance decreases as the magnification of objective lens increases. What will be the answer? The working distance decreases as the objective lens increases. What will be the answer? Yes, it is true. The working distance decreases as you increase the magnification. The high power objective lens has to be much closer to the specimen than the low power objective lens in order to focus. Working distance is inversely proportional to the magnification. Ninth question is, which of the following are low power lens? Low power lens. Yes, 4x is a scanner, but 10x is known as the low power lens. Which part of the microscope contains the shutter that regulates the amount of light entering the lens system? Which part will regulate the amount of light entering in the lens system? Yes. The condenser, the eyepiece, the stage, or fine adjustment knob? Yes, the condenser. The condenser is equipped with an iris diaphragm, a shutter controlled by a level lever that is used to regulate the amount of light entering the lens system. Above the stage and attached to the arm of the microscope is the body tube. A microscope is set to 10x eyepiece and 40x objective. What will be the total magnification? This is the very same question as we have seen earlier. What will be the total magnification? Yes, 400x. When focusing, it is best to start with the lowest power. Is it true or false? Yes. Whenever we start for the microscopy, we should use start from the lower power, so that we can have a overall scan about what specimen we are looking for, and then we should proceed to the higher power. Next question is, which part of the microscope are objective lenses attached? With which part of the microscope the objective lens are attached? Yes, it is nose piece, as we have seen in the video. An objective lens is the most important optical unit that determines the basic performance or function of an optical microscope. Which of the following liquids can you use to make a wet mount? Which of the liquids Yes, water. Now, what is the wet mount? A glass side holding a specimen suspended on a drop of liquid like a water for microscopic examination, and also a specimen mounted in this way. The next question is, when viewing a specimen, start focusing with the fine adjustment knob and then fine tune using the coarse adjustment knob. How will you proceed to? Yes, it is false. First, you have to do a coarse adjustment, then only for the fine focusing, you have to use the fine adjustment knob. Next question is, to obtain total magnification, you should, what you have to do with the magnification? The magnification of eyepiece to the objective lens power. What to do? How do you calculate? Yes, multiply. We have to multiply the magnification of eyepiece and the objective lens to obtain the total magnification. Now, next question is, match the part with each function. So this is the revision of our whole practical class by this one question. Now, how will you match? What will be the match of these parts? Here we can see that from A to J, these are the left side. They are the parts of the microscope, and in front of them, the functions are given. So what will be the correct match? You can answer one like A for eyepiece, and against that, which number means what is the function of eyepiece? Yes, who will answer?
This is our third revision. Once video was shown, we have taken up questions even, and now this one is just the revision. So all of you know it very well.
This is the revision. We have seen the whole practical class and the video. We have gone through the questions for the same. So you must be knowing the functions. Yes, J4. Base is functioning as the supporting system, support the microscope. Yes, F is sharpen, used for sharpen the image, means for fine focusing. The stage clips for holding the slide in place. The stage to support the slides, yes. Diaphragm to regulate the amount of light. E, the coarse adjustment knob will focus the image. Yes. Okay, now we see the answers one by one. So the eyepiece, eyepiece, the function of eyepiece is to magnify the image. The body tube, it causes the body tube will reflect the light towards the eyepiece. The arm is to support the body tube. The stage clips to hold the slide in place. Coarse adjustment, which is used to focus the image. Fine adjustment tube knob for the sharpening the image. Then diaphragm, which will uh, regulate the amount of light. The stage will causes holding of, supporting the slide. The light source will that will reflect the light towards the eyepiece, and the base will support the microscope.
So almost we are coming to the end of this demonstration class. And though we have here one slide on identifying the parts and to put down the use of 3, 4, and 10, we will not ask you here what is the correct answer. But remember, when you come here as offline, you will be issued journals. So in the journal, you put down the answers to the questions, because we learn in detail. So you will be able to put down the answers, and a microscope parts, even you will be able to label it properly. So we go ahead with still 2 more questions. So which oil is used for studying under oil immersion lens? Anybody can put down the answer in the chat box. What oil is used? Yes, yes. So the answers are correct. You can use cedar wood oil. But remember, practically, we are not using it in our practical class. It is just the paraffin oil that we are using. Why we are not using the cedar wood oil? Number one, it is costlier than paraffin oil. And second is at a long term, it damages, destroys the lens of the objectives. So paraffin oil that is used for oil immersion lens and not cedar wood oil. And the last question, what is the use of oil when viewing through an oil immersion lens? The last question, what is the use of the oil? Why do you need to put a drop of oil on any object? So here is the answer. Immersion oil, it contributes to two characteristics of the image viewed through the microscope. Which are these two characteristics of resolution, and second is the brightness, by which it helps to identify the structure very easily. So these characteristics are the most critical under high magnification. So it is only the higher power short focus objectives that are usually designed for oil immersion. So now we can take up if anybody has any query. We have still time. We can have some time for allowing questions. Or if you don't have questions, then you can even discuss anything you want to clear your doubts in the practical class when you join offline. Is it correct? So nobody now want to put any comment in the chat box for any questions? So have you understood something on the microscope? That question would be easy to answer, okay? It is either yes or no. Very good, very good. Okay, okay. Still some more important point for the microscope is its maintenance. You need to take care. It is very costlier. So when you want to handle it, you must be taking enough care. And what are the points that you need to know about taking care when you are in the practical class? Who can answer? Please, you put down in the chat box. Or you can mute your mic. Yes, I will allow you. I will allow you and you answer on the mic. What are the precautions you should be taking for using a microscope in proper handling? Okay, when you are using the oil immersion lens, how do you clean the oil immersion lens? Yes, very good. Rutam has answered one of the precaution that is very good. You cover the microscope when you are not using it. One number, that is one of the first one. What is the second one? Before covering, you need to clean the oil immersion lens. How do you clean it? With your apron? With tissue paper, not with cotton. Cotton is wrong. Please, you use your tissue paper, not only tissue. Tissue can be anything. Something removed from the body is also tissue. So you cannot write tissue. It should be soft tissue paper. Soft tissue paper. Some of the students, they would advise again one liquid, that is, yes, tissue paper is correct. They will advise you to put xylene. That is wrong. You are damaging the lens with xylene. So don't use even xylene. It is soft tissue paper that you can clean the oil immersion lens and then you cover it. Why do you need to cover it? To avoid dust. And when you are shifting one microscope from one table to another table, please you see that with one end you hold the handle and with the other hand you hold the base so that there is no tilting of the microscope and you carry it in just the horizontal manner. Okay, so this is enough for the day, and plus every few months you need to clean the microscope. That is also with tissue paper, and the cover that you are using for it to cover the microscope should also be washed regularly. So we end our practical class. Okay, good day to everyone and hope you have enjoyed the simple class that is on microscope. પણ આમાં end meeting કરવાનું હોય છે તો આ કરું હા end meeting એટલે એ લોકોની attendance લખાઈ જાય એવું છે